Perhaps you've heard of our recent terrestrial and seagrass BioBlitz events on Calvert Island, where ecologists scoured the landscape to identify as much biodiversity as possible in the span of just 1 week. These events are more than just a giant scavenger hunt, though. As the specimens begin to pour in, there's a lot that goes on behind the scenes to catalog every ant, beetle, lichen, plant leaf, and strand of seagrass. By creating an accurate snapshot of what kind of living organisms inhabit this unique place, we can track biodiversity responses to environmental change over time and understand how biodiversity is connected across space.
Their main importance comes as a way of trying to discover what's around us. So, for example, it could have a practical value for conservation. You can find endangered species or discover which habitats we might want to be giving different kinds of protection to. But it's also kind of like real, true blue sky sort of astronomy type discovery. Just as finding a new planet or other orbiting body is exciting, it's exciting to find a plant or animal that you didn't know was there, maybe even something new to science.
Survey and collection locations are recorded by field technicians on every outing using a GPS to allow us to compare our findings to different areas or at different times. Then comes the microscopes.
But even just, sitting down with a single specimen, if it's a challenging one, just to sort of work through the keys and sort of go through that process of puzzling out or figuring out what you've got, I like that challenge as well.
So, and also, we collect hundreds or thousands of spiders. And sometimes when you're doing that, there's one individual of that species. So, others, there's 200 of it. So it's those onesies that are usually the really cool and interesting ones.
Many specimens will be processed for DNA barcoding, where every species is given a unique barcode, just like the barcodes that get scanned whenever we buy things at a store. The resulting DNA sequences are then added to the open access Barcode of Life Database project at the University of Guelph.
We just essentially grab one of the legs. I put it in the vial and then I put a little bit of 95% ethanol in here. Fill it up, and that's all you really need, one leg, in order to get DNA from that.
While many collectors would prefer to release their finds rather than kill them to sample DNA, doing this now might mean not having to down the road.
In the future, we may not have to kill the specimens at all. We can sample the water, and then magic, they're all identified through the genetic material in the water.
In the plant tent, once samples have been identified, they are laid out and pressed between sheets of cardboard to be sent off. The real fun happens when the results of the DNA samples are returned months later. Things are not always what they seem.
I will be very surprised if we don't have some species that are discovered that are brand new to science. And to think that in Canada, that you can still do that you can gather together a group of a couple dozen experts, turn them loose, and they'll come up with new species to science, I think that's a pretty special thing.